You know, in modern biology, we've got this incredible challenge. We generate so much data. Sometimes we need to zoom in and focus on a single tree, and other times we really need to step back and understand the entire forest. Today, we're talking about making sense of that massive amount of data from omics environments, and it all comes down to learning which lens to use. So let's dive in.
So picture this. You've just wrapped up a huge experiment. You're sitting on a dataset with expression levels for, say, 20,000 genes across all of your samples. I mean, that's an amazing amount of information. But where do you even start? It's that classic problem, right? You're drowning in data, but you're starving for actual information. Honestly, it's like staring at a giant mountain of data. And if you want to find the really valuable signals hidden inside, you can't just start digging anywhere. You need the right tools, the right lenses to see what actually matters. And today, we're going to look at the two fundamental lenses we use to analyze this mountain. All right, let's start with our first lens. This is the one-by-one approach. It's often the very first tool that researchers reach for, and its formal name is univariate analysis.
So what does univariate even mean? Well, it's actually pretty simple when you break it down. Unit means one, right? And variate means variable. So you're literally just analyzing one variable, in our world, one gene at a time. It's a very focused, methodical way to start sifting through everything.
To make this super clear, let's stick with that analogy we started with. Imagine your entire dataset is a massive forest. Using a univariate approach is like walking through that forest and checking one single tree at a time, just to see if it's maybe taller in one part of the forest compared to another. And here's the key thing you got to remember about this method. The whole process works by taking just one gene, comparing its expression between your groups, say your healthy group versus your diseased group. You get a result for that one gene. Then you set it aside, and you do the exact same test for the next gene, and the next, and the next. Each one is treated completely independently.
So, what's the result of doing all that? Well, you get this. When you run a separate, independent test for every single gene in your dataset, you end up with just an enormous number of results. We're talking more than 10,000 p-values. And that creates a whole new problem. How do you know which of these are really significant and which just looked that way by pure random chance?
And that leads us to something that is, well, it's absolutely non-negotiable if you're doing this kind of work. Because you've run so many tests, you have to apply something called multiple testing correction. It's basically a statistical adjustment that helps control for all those false positives you're bound to get. Seriously, it's essential in omics. So what are these tools? You've probably heard of them. These are the classics in that univariate toolbox. If you've got two groups to compare, you'll probably use a t-test. If you have more than two, you'll use something like ANOVA. And hey, if you want to see how a gene's expression changes with a continuous variable, like age, you'd use linear regression. Each tool is built for a specific one-on-one kind of comparison.
Okay, so that's the one-by-one approach. That's looking at the trees. But what if you don't care about a single tree? What if your question is about the shape of the entire forest? Well, for that, we need a completely different lens.
And this is where our second lens comes in. It's called multivariate analysis. And, you guessed it, the name tells you everything you need to know. Multi means many. Here, we are analyzing many variables, or tons of genes, all at the same time. This brings us right back to our forest analogy, because it really is the perfect way to think about it. We're no longer focused on any individual trees. Nope. Instead, we're taking a bird's eye view. We're studying the entire forest to see how groups of trees grow together, what the overall landscape looks like, and what major patterns emerge from the whole system. So here's the magic of this approach, the real key feature. It's not just about what genes are up or down, it's about the relationships between them. It asks questions like, how do these thousands of genes behave together? Do certain genes always go up when other ones go down? It is all about interdependence and how things vary together. And the tools in this toolbox, they have totally different jobs. You've got methods like PCA, which are fantastic for just visualizing the biggest patterns of variation in your data. Then there's clustering, which helps you find natural groupings, like do these samples seem to belong together? And then you have supervised methods like PLSDA, which you use to build models that can actually classify your samples into groups you already know.
All right, so now we've got our two very different ways of looking at this mountain of data, right? One for the trees, one for the forest. Let's really put them head to head to make the difference totally clear.
I think this is the most practical way to think about it. Like, forget the theory for a second. What do you actually get at the end of the day? Well, a univariate analysis gives you a list.
a ranked list of individual genes that are significantly different between your groups. But a multivariate analysis? That gives you a map, a visualization like a PCA plot that shows you how all of your samples relate to each other based on their entire genomic profile. A list versus a map.
So let's just break it down one last time with this table. It really nails the difference. The focus? One at a time versus many altogether. The goal? Finding individual differences versus finding big global patterns. And the output? A long list of p-values versus a visual map or a classification model. See? They're two fundamentally different approaches, designed for two different kinds of questions.
So after all of that, you're probably thinking, okay, so which one's better, the univariate or the multivariate approach? And you know what? That's actually the wrong question to ask. It's not about which one is better or worse. It's about picking the right tool for the job you need to do.
And this right here is the most important thing I want you to walk away with today. The statistical method you choose has to be driven by your scientific question. There's no such thing as a one-size-fits-all solution here. Your goal always dictates your tool.
So, the next time you find yourself staring at that mountain of omics data, just ask yourself one simple question. Am I trying to find the single tallest tree in the forest, or am I trying to understand the overall shape of the forest itself? Your answer to that question will tell you exactly where you need to begin.